paper.
Okay, this is Mr. Brechko back with Vivian. Say hello, Vivian.
Hello, Vivian.
And Vivian, what are you working on?
Paper.
Cool. And I am going to be explaining meiosis.
Yeah, this is your meiosis.
All right, so we're going to go through the basics of meiosis, and we're going to go through each of the stages of meiosis and how they work. So meiosis is going to start out with meiosis 1. Meiosis 1. And this is the first cell division. All right, and so we're going to start out with interphase 1. And interphase 1, you're going to have your cell. And again, this is going to be occurs in the gonads.
Yep.
Which are the testes and the ovaries. All right, so, and humans anyways, that's where it occurs. So you're going to have your cell, and you're going to have your nucleus in your cell, and you're going to have all of your DNA unraveled in the form of chromatin.
Oh, my paper.
And the DNA is going to replicate.
My paper was cutting.
Do you want to go down and get it?
Oh, you get it.
You want me to get it? Can you say please?
Please?
Okay. Oh, it's too far. I can't get it. You have to go get it.
No.
Yeah.
I'm going to cut this.
Oh, yeah, you can cut the next one.
Okay.
All right, so that's important so that when the chromosomes, which originally were just single-stranded, are now going to have their identical strand that makes up the other half of the X. And remember that these are held together by centromeres right in the middle there. All right, so now we're going to go to prophase 1. And in prophase 1, I'm going to draw the cell a little bit bigger here. And we'll draw the nucleus kind of starting to dissolve. And our chromosomes are going to form. So the chromatin is going to wind up to form chromosomes. So what I'm going to do is I'm going to draw 3 pairs of homologous chromosomes. So we're going to assume that this organism has 3 chromosomes, unlike humans, which have 46 chromosomes, or sorry, 23 pairs of chromosomes. This organism is only going to have 3 pairs of chromosomes. I'm going to draw them using red and black. So we have a big chromosome, which will be chromosome 1, and we're going to have a medium-sized chromosome, which will be chromosome 2, and a small chromosome, which will be chromosome 3. All right, so we have 3 pairs of homologous chromosomes.
Paper.
All right, so that's prophase 1. Just to highlight, there's pair 1, here's pair 2, and here's pair 3. And prophase are just kind of randomly arranged inside of the cell, so it doesn't really matter where they are. All right, so if you remember, PMAT, the next phase after prophase is going to be metaphase 1. What's going on, Viv? Can you say prophase 1?
Prophase 1.
Can you say metaphase 1?
Metaphase 1.
Good job. So metaphase 1 is a really important phase for meiosis. Okay? So what's going to happen is our spindle fibers are going to attach to the chromosomes. And they're going to line them up along the middle of the cell. And all the chromosome 1s are going to line up. And they're actually going to join together to form a structure called a tetrad. Okay? Tetra meaning 4. Okay? And then chromosome 2, these guys are all going to line up, forming another tetrad. And then chromosome 3, these guys are going to line up, forming a third tetrad. All right? And this process is called, or this moment in cell division is called synapsis. Okay? In metaphase 1, when tetrads form. Okay, and the tetrad is 4 homologous. Did you fall out of your chair again? A tetrad is 4 homologous chromatids joined at the centromere. Okay, so this is where the first kind of cool thing happens. Are you talking? The first kind of cool thing that happens is, so I guess I should say that sexual reproduction adds variation to a species. And meiosis is kind of where a lot of that variation happens. You want to sit up here so you can see what's going on?
Alrighty.
Yeah. So one of these sources of variation are that when the tetrad forms,
Look at that.
the DNA can actually swap. So look at chromosome 1, and what's actually going to happen here, and I'll show it in green ink, is the ends of these chromatids are actually going to swap DNA. So there's going to be some red on that black one, and there's going to be some black on that red one.
Yeah.
And they actually swap DNA. So the genes that were originally on this,
Black.
the genes that were on this originally on this red chromatid are now on the black chromatid, and vice versa. This is going to result in mixing up traits. Okay. Oh, careful. Don't touch it. All right, so that's kind of metaphase 1. We have synapsis, we have the tetrad, and we have crossing over. And crossing over is where homologous chromatids swap DNA. Oops. And this is going to increase variation. Vivian, what do you think? Good? Yeah? Can you say variation?
Variation.
Can you say crossing over?
Crossing over.
Can you say synapsis?
Synapses.
Can you say tetrad?
Trap.
Say good job.
Good job.
All right, so the next phase, if you remember, is going to be anaphase.
Yeah.
And this is going to be anaphase 1.
Two,
So in anaphase 1 is where the chromosomes are going to be pulled apart to the poles of the cell. So I'm going to draw the cell being kind of stretched out.
Yeah.
And we'll draw the spindles here, pulling them apart. And this is where the second really interesting thing happens.
apart.
That's right. All right, so
Yeah, they're all gonna be pulled apart.
good job. So our first chromosome, I'm going to draw getting pulled apart, and 2 tetrads are going to be pulled to each pole.
Yeah.
And it's completely random how these 2 tetrads get pulled. So you can see in this case, we had 1 black chromatid go to the left side and 1 black chromatid go to the right side.
At the bottom.
And likewise with the red. All right, on the medium chromosome,
To the bottom.
I'm going to draw 2 black going to one side, 2 red going to the other side. And on the small chromosome, I'm going to draw it the opposite. But it doesn't matter, and it's random every time. Yeah, I'm going to have you go run around the table 4 times.
Okay.
Okay?
4 times.
All right? So this process is called disjunction, which is the pulling apart of the chromosomes. Now, occasionally, you did it? All right, now I want you to go roll on the ground 10 times.
Like this?
Like a log. Not a somersault, because you'll bang into something. Roll like a log. But roll over here where it's open, where you won't bang into stuff. All right, so occasionally something kind of, the other interesting term here is independent assortment. And this is the idea that each chromosome I should say each, I'm going to erase that. I should say each tetrad splits randomly.
Time.
Really?
Times.
All right, can you cut out 4 pieces of paper and tape them together?
Yeah.
Okay. Different colors, though. You can put your oranges in the garbage. So-
But I don't need to.
Okay, so each tetrad splits randomly, and this is going to increase variation. This is how we wound up with the red and black here, and then black and red, and then red and black. That's how we did that. And so this is going to increase variation. Just like our crossing over increases variation. Okay, now a third and interesting thing can happen here, and that is called non-disjunction. And non-disjunction is a failure of the tetrad to separate evenly. So what would happen in this case is, I'm going to do it with chromosome 3, I'm just going to erase that, is only one red would go to that side and the other red would go to this side, resulting in this package of chromosomes here having 3 copies of chromosome 3, where on the left side, which I'll do in blue, you only have 1 copy of chromosome 3. And that's going to have, what that's going to result in is eventually, when you have a sperm or an egg, a one-end cell, it's going to have that extra copy over here, and that's going to result in maybe trisomy. If it happens on chromosome 21 in humans, you're going to have trisomy 21 or Down syndrome. Good job, but that's 2 pieces. You got to do 2 more pieces.
2 more?
Yeah. Then you can tape it to the. Yeah. Then you can tape it to the chalkboard and draw circles around it. So that about wraps up the interesting stuff for anaphase 1. Telophase 1. Can you say telophase, Vivian?
Telophase.
Good job. Now the cells are going to pinch off. And over here you're going to have your, let's see, how do we do this? Black chromosome here with a little bit of crossed over chromosome there. And on the other side you're going to have your red chromosome with the crossed over black chromosome. And then in this cell for chromosome 2, you're going to have your 2 medium-sized black ones over here, your 2 medium-sized red ones over here. And then a single red one here, and our triple over on the cell on the right-hand side. So this is just like what happens in mitosis. The cell pinches off. Or else you'd have the cell plate form if it was a plant cell. All right, so that's the first division. Now you have 2 cells. So now we're going to have prophase 2, which is obvious. You just kind of have your 2 cells getting ready to divide again. So that's kind of like boring. We'll just skip that. And then we're going to have metaphase 2. And so we're happening in 2 cells now. So here's our first cell and our second cell. And we can draw our spindles. I can do mine in gray. You would have centrioles at the end of these cells if it was animal cells. I'm going to draw them just to keep it simple.
Daddy,
What's up?
I'm cutting all the paper.
Good job. So I'm just drawing the chromosomes lining up in the middle. Okay, and this is really just like mitosis. So the next step is going to be anaphase 2.
Ta-da! Daddy,
Yeah. How's it going? Oh, wow, that's a good pile of paper. Are you going to tape those together in a chain? That's a good idea. So in anaphase 2, in each of these cells, I'm going to draw them stretched out. We'll draw our spindles again. And now we're going to draw our chromosomes getting pulled apart again. We'll draw black going to this side, and then the red chromatid or chromosome with the little crossing over going to that side. And it doesn't matter which side you draw these going to, once again, because it's random. It's the whole idea of independent assortment. Each chromosome is split independently. So here I'm going to draw my two medium-sized black ones being drawn to the ends. On the other cell, we'll draw the two medium-sized red ones being drawn to the ends. And then over here, we'll draw one lone red one being drawn to the end. And over here, I'm just going to draw, make it interesting, a red and a black going to one side, and a black going to the other side. And notice here, there's nothing there. So here's our two opportunities for something unusual to happen with the zygote, and that either there will be an extra copy of a chromosome or it'll be missing a copy of a chromosome. Okay. So then in telophase 2,
Daddy,
yes.
can I put it in the water?
What do you want to do?
Play with the arrows?
No, you can't play with those. Those can break. But you can go to the sink in the back there,
Yeah.
and you can play in that water a little bit.
And just,
But you'll have to move your chair. You have to drag your chair over there.
what about break anything?
I don't think you'll break anything. Well, I thought you were going to tape your paper to the board and draw circles around it. All right, so in telophase 2, the cells are going to pinch off. Should have some interesting sound effects here. All right, Viv, can you wait to do that for a couple minutes? No? All right, go ahead. Drag it back. Go fast. Go fast. Well, she drags a chair. I'm going to draw these chromosomes. Oops, that should be a red one in this cell. Just copying from up above. This is going to be my big red one with a little black tail on it, followed by a medium-sized black one and a missing chromosome there. And then we're going to have a big black chromosome here with a red tail on it, chromosome 1. And then over here, we're going to have this guy. And then down here, we're going to have our two red medium-sized chromosomes, little guy. There we go. Okay. So this results in basically our 4 gametes, or sex cells. Okay, so in human males, this is going to become 4 sperm cells. And human females, one of these will become an egg cell, and the other 3 will become polar bodies. All right, and it's completely random in fertilization. Randomly selects a gamete to form the zygote. Okay, so between randomly selecting one of these gametes and the independent assortment that occurred during both anaphase 2 and anaphase 1, and the crossing over that occurred in metaphase 1, we have lots of opportunity for variation in the offspring. And that's what meiosis gives us. It gives us, again, as a summary, it gives us 4 end cells with half the normal number of DNA from an original 2-end cell. And then fertilization, those end cells can then join back together to form a 2-end cell, although it wouldn't be from the same organism. And finally, just to compare our end cells right here to the original 2-end cell, notice that we only have 1 copy of chromosome 1, 1 copy of chromosome 2, and 1 copy of chromosome 3, whereas originally we had 1, 2 copies of chromosome 1, 1, 2 copies of chromosome 2, and 1, 2 copies of chromosome 3, and they were replicated. Okay, because remember that two sides of the X are identical to each other. So I think that about wraps it up for meiosis in detail. Good luck. See if I can stop this recording for you.